comparing the data between the placebo and phage for the sake of brevity and time. In terms of safety, of the 64 patients that were exposed to phage treatment, only one case of mild exfoliation was reported. It resolved quickly by the next clinic visit in 4 weeks. And in terms of efficacy, the phage arm recorded the highest treatment efficacy, which was 57% response, followed by placebo and phage plus sal acid. In terms of mechanism, the phage group did show a lowering of C. acnes levels, which was very validating in terms of the MOA, whereas placebo C. acnes levels went up and phage plus sal acid levels also did not go down. In terms of microbiome diversity, we saw a really interesting phenomenon where the phage group saw a significant increase in diversity compared to placebo or phage plus sal acid. And I'm sure you're seeing this pattern now where the phage plus sal acid, 2%, did not perform as well as the phage alone, which tells us, A, that the phage alone is, working to cure acne. But the second point was that we realized that the sal acid MOA is actually orthogonal to the phage. So sal acid is an antikeratolytic. It opens up the pore, floods it with oxygen, and as a result, the C. acnes, which is anaerobic, stops growing. Now, phages need an actively dividing C. acnes in order to infect it and continue its life cycle. And so adding high amounts of sal acid was really sort of halting that march of the phage into the pore. And so I'll share with you a little later how we've solved that problem in formulation. But in conclusion, we found that the phage alone, phage treatment, works to reduce the acnes level, which results in an improvement in acne and an increase of microbiome diversity. And although this pilot study had a limited N, we are seeing an efficacy signal from the phage arm in 8 short weeks.